The night is soft around you. Your body lies still, quiet under a blanket of breath and bone. And beneath your skin, beneath your thoughts, something ancient stirs. Not loud, not sharp, but steady. It is the hush of instructions humming inside you like a lullaby known before birth. This is your code. Each of your trillions of cells holds a copy of this quiet manuscript. It is written not in ink, but in a language of life. 4 letters, endlessly rearranged. A, T, C, and G. The smallest alphabet with the largest meaning. These letters don't form words the way human language does. They weave something more mysterious. They shape you. You do not need to understand these letters for them to work. Like breathing, they happen without asking. Right now, as you listen, your cells are following that code, whispering their instructions in the dark. Grow a new skin cell here. Keep the heart beating. Make the chemical that helps sleep wrap softly around the brain. All of this happening in silence beneath your awareness. DNA, they call it. Deoxyribonucleic acid. But tonight, let's not call it a chemical. Let's call it what it feels like, your body's poetry. A long, spiraled letter, folded tight inside the nucleus of every cell. Unseen, but always present. Like wind in a field, like memory in a dream. This is what connects you to the very first living things on Earth. They too wrote in DNA, and over billions of years, that script has been copied, changed, refined, and passed down, until one day it arrived in you, not broken, not perfect, but endlessly complex. Let yourself drift deeper now into the spiral. The DNA inside your cells is coiled with great care, like a sleeping serpent of code. If you could take the DNA from just one cell and stretch it out, it would be 2 meters long, longer than your height, and yet it folds inside something so tiny, thousands of times smaller than the width of a single hair. Imagine that folding, like origami made of meaning. These spirals form chromosomes, tightly packaged bundles that hold your story. You have 23 pairs of these bundles, one set from your mother, one from your father. Together, they shape everything from your eye color to your immune system to how your body responds to certain foods or medicines. It's a kind of music, really. A double helix, 2 strands twining together like twin violin strings. The letters on one strand pair with those on the other, A with T, C with G, always. A faithful duet repeated again and again across your entire body. But this song isn't simple. It holds about 20,000 genes, each one a specific instruction, a recipe for a task your body needs to carry out. Some genes are busy all the time, like those helping your cells divide and grow. Others sleep, only waking under special conditions, like stress, sunlight, or the scent of something familiar. Think of genes as paragraphs in your body's instruction manual. Some paragraphs are short and precise, others long and poetic, and some are cryptic, filled with code we still do not fully understand. This book, this inner book, never leaves you. You carry it into every room, every conversation, every dream. But here's the quiet miracle. Not every cell reads the whole book. A skin cell reads only the part about being skin. A liver cell reads its own chapter. A neuron in your brain flips to the section about thought and electricity and feeling. Each cell chooses what to read, and then, almost magically, it follows the recipe. The gene is read, copied into a message, and sent into the body of the cell. The message tells the cell to build a protein, a tiny tool or structure made of atoms. Proteins are the workers of your biology. They form the scaffolding of your muscles, the pigments in your eyes, the enzymes in your digestion. Proteins are what genes become when they are read aloud. And every second, your body makes millions of them, following those 4 letters, A, T, C, and G, over and over again, like a lullaby that never forgets the tune. Let's pause here. Let this settle. Because this is not a fast thing. This is not the clicking of machines or the hard logic of computers. It is soft, slow, winding, like a child's breath. The cells do not panic. They do not rush. They follow the dance they've followed since the first spark of life, a kind of cellular memory older than any language. And though we try to understand it, there are still things we don't yet grasp. We know that certain genes are tied to certain traits, to certain illnesses, but many genes do not act alone. They work in teams, in networks, in feedback loops too vast to trace fully. One small change in one letter can shift the balance. And sometimes it breaks the music. A wrong letter, a single misplaced nucleotide, can change a word, distort a protein, silence a gene. When this happens in certain places, the body may falter. These are mutations, changes in the code. Some are harmless, some are even helpful, but some lead to inherited diseases. But not yet, not tonight. Tonight, we stay in the wonder, because most of the time the system works. It copies, edits, repairs. Your body catches many of its own mistakes. There are even tiny repair crews, proteins designed to recognize when DNA is damaged and quietly fix it. Like gardeners trimming stray branches or monks copying sacred texts, careful to keep the meaning intact. You've never seen them, but they've seen you. They work as you sleep. And when you wake, your cells are still humming with that code, still spinning the molecules that keep you alive, still following the instructions first written long before your name was chosen. This is what makes life so deeply shared and so deeply yours. You and every other human on this planet share 99.9% of the same DNA. That last 0.1% accounts for all your uniqueness, the shape of your hands, the curve of your voice, the way your brain sees color or solves puzzles. A whisper of difference in a song we all sing, and yet that tiny difference can mean everything, in joy, in illness, in how we live. So what happens when we read this book and find an error? What happens when someone is born with a single flaw in their code, one that leads to pain or weakness or loss? Can we edit the story and can we fix the letters? Can we cure inherited disease, not just treat it, but undo it, remove it from the line of life? That is where we begin next. But for now, you rest. Feel your breath. Know that deep inside your cells are reading. They are writing. They are repairing. They are trying always to keep you whole. And that is a kind of love, isn't it? Your body loving you in silence. Even when you sleep, even when you forget, even when the code is not perfect. Tomorrow we will follow these threads further, to the moment when the story begins to tangle, and the dream of untangling begins to feel real. Your body is still, but your story is moving. Not the kind of story made of memories or names, but one etched deep into your cells, a story written long before you took your first breath. Tonight, you drift back, not just through time, but through generations. Each moment of your existence is woven from invisible threads passed down silently from those who came before. You are not just one life, you are thousands. Float now gently through the centuries. Imagine your parents as children and their parents as children, and the line stretching farther back than your eyes can follow. What they passed on was more than stories, more than heirlooms. They passed on genes, tiny sequences of DNA carrying instructions, tendencies, and patterns. These genes flow through you now. Some you'll never notice, and others shape you in quiet, surprising ways. Inside every cell of your body are those 23 pairs of chromosomes, those bundles of DNA we met in the last chapter. Each pair contains one chromosome from your mother and one from your father. Imagine them now exchanging gifts. One thread from her, one thread from him, woven together into a tapestry that becomes you. This process is called inheritance. It's how traits pass from parent to child. Not just your eye color or the shape of your jaw, but how your body makes blood, processes sugar, or responds to infections. Even how your mind works, the balance of your neurotransmitters, your ability to focus, your patterns of sleep, may be shaped in part by the code you've inherited. But inheritance is not destiny. It is possibility. A blueprint, not a prophecy. Let's pause here. Breathe with the knowledge that you are part of something old. Your cells remember what your mind cannot. When your mother carried you, she carried not just your body, but the genetic material that would shape your future. Even inside her, your egg cells or sperm cells were already beginning to form with their own copies of this ancient code. You were once inside your grandmother, and a part of your grandchildren already lives inside you. Like Russian nesting dolls of biology layered across time, the way traits are passed is both elegant and mysterious. Some traits are dominant, bold, assertive, likely to appear in each new generation. Others are recessive, quiet, patient, often hidden for years before surfacing. Take, for example, the color of your eyes. Brown is typically dominant. If one parent gives you a gene for brown and the other for blue, brown usually wins, not out of aggression, but efficiency. But sometimes two recessive genes meet. And then the quiet traits step into the light. This is how you might inherit your grandmother's green eyes, or your great-grandfather's curling hair, or an ability to roll your tongue that skips a generation before reappearing in you. It's a dance of combinations, and chance plays a part, but so does biology. Now, drift into the concept of carrier status. You may carry a gene for something you do not have. You may walk through life never knowing it's there. Like a folded paper in your pocket, silent, unopened. But if someone else also carries the same folded paper, and you pass them to the next generation together, the message inside may unfold. This is how some inherited diseases appear. Not because one person did something wrong, but because two quiet threads aligned. Cystic fibrosis, Tay-Sachs disease, sickle cell anemia. These are just a few conditions that may appear when both parents carry a hidden variation in the same gene. The child may receive two altered copies, and with them a different kind of path. It's not a flaw, it's not a punishment, it's a twist in the thread. But not all traits come from just one gene. Many are polygenic, shaped by the interaction of many genes together. Height, for instance, is influenced by hundreds of small factors. So is intelligence. So is your risk for diabetes or heart disease. The more complex the trait, the more genes are involved, and the harder it becomes to trace. Imagine it like a woven blanket, not made of one bold thread, but of countless fine strands, twisting together to form the pattern you wear. This is why inheritance is never black and white. It's shades and shadows, ripples and echoes, and the environment too plays its part. How you live, what you eat, the air you breathe, the stress you carry. Genes are not just instructions, they're responsive. They can be turned on or off by the world around you. This brings us to epigenetics, the science of how experience touches inheritance. Imagine your genes as a library. Epigenetics is like putting bookmarks in that library. Some pages opened wide, others gently closed. Stress, trauma, love, starvation, kindness, they all leave bookmarks. They can silence a gene or amplify it. And sometimes those bookmarks are passed to the next generation. This means your experience may affect not just your life, but your children's, and maybe their children's too. It is both beautiful and humbling. Your healing becomes their foundation. Your pain, if unaddressed, may echo forward. But nothing is final. Even bookmarks can be moved. In this endless web of connection, you carry more than just human threads. Your DNA also holds remnants of ancient viruses, small genetic fossils tucked away, memories of battles fought by your ancestors, their immune system adapting to survive. Some of these viral fragments now help you. One helps build the placenta in pregnancy. Another may assist your body in detecting danger. You're not only your family, you're every life that shaped your lineage, and your lineage stretches wider than you know. If you look far enough back, we all share ancestors. You are 9.9% identical to every other human on this planet. That 0.1% variation tells the story of your ancestry, where your people walked, what climates they lived in, how their bodies adapted to survive, your skin, your enzymes, the way you process milk or sunlight, all of it shaped by the lands your ancestors once called home. You are not separate, you're a note in the ongoing song of life, and it sings in your blood quietly. So now, as you rest, you might wonder how much of you was chosen, how much was inherited, and how much still can be shaped. These are the soft questions that genetic science holds. And here is where healing enters the story, because if we can trace the threads we inherit, might we also learn how to mend them? What if you carry a gene for an illness, but science finds a way to gently correct it? What if the legacy you pass on could be edited with care so that your child inherits only the light? What if we could whisper into the threads and say, "Be well"? That is the path we begin next. We've seen how the code is passed. Now we begin to notice when the pattern changes, when something breaks, when a single letter brings more than difference, it brings suffering. But even then, the story does not end, because humans too adapt. We observe, we learn, and we try, with gentle hands and curious hearts, to repair. Drift now in the quiet understanding that your story is not only ancient, it is ongoing. And perhaps, just perhaps, its next chapter holds something new. The night stretches long and quiet around you. Within your body, countless rhythms continue. Cells divide, DNA coils, proteins unfold like origami into shapes they've held for generations, the music of life soft and steady. But sometimes the melody wavers. One note slips. One letter of the ancient genetic script is out of place. And in that tiny moment, something changes. Not all changes bring harm, but some do. And that's where we begin tonight. In the quiet tangle of threads, you are made of code. Inside every cell, DNA holds instructions written in 4 letters, A, T, C, G. They arrange into genes, and genes guide how your body grows, repairs, breathes, and becomes. Each gene is like a recipe. It tells your body how to build something, an enzyme, a pigment, a structural protein. Now imagine what happens when a letter in that recipe is switched. Just one. An A becomes a T, or 3 letters vanish completely. That tiny shift can cause a ripple, like a pebble tossed into a still pond. That's called a mutation, a single small variation in the DNA code. It can be silent, barely noticed, or it can change everything. Let's imagine your genome as a woven tapestry. Millions of threads knotted in patterns passed down from your ancestors. Most knots are tight, well placed, but some, they twist, snag or fray, and in those snags inherited diseases begin. Some illnesses arise from a point mutation, a single letter change in the DNA. Others come from bigger shifts, deletions, duplications, or misplaced sections of a chromosome. Think of it like a manuscript. You can change the meaning of a sentence by swapping one word, but lose a page and the whole story changes. One of the clearest examples is cystic fibrosis. This condition begins with the deletion of just 3 DNA letters. That's all, but those 3 missing letters change the way cells manage salt and water. The result? Thick, sticky mucus in the lungs and digestive system. Chronic infections, difficulty absorbing nutrients. Is not a punishment. It's not because someone did something wrong. It's simply the result of a thread woven with one loop missing. Or take sickle cell disease, caused by a single change in one base of one gene. That shift alters the shape of hemoglobin, the protein in red blood cells that carries oxygen. Instead of flowing smoothly, the cells stiffen and curve into crescent shapes. They get stuck, break, cause pain. One tiny change, one lifelong journey. These diseases are monogenic, caused by mutations in a single gene. But not all inherited illnesses are so simple. Some arise from polygenic causes, where many genes, each with tiny effects, combine like whispers into something louder. Conditions like heart disease, type 2 diabetes, or schizophrenia often don't come from one clear source. They're both shaped by dozens, sometimes hundreds of genes. Each gene adds a brushstroke, and together they paint a risk. This is why inheritance can be so difficult to trace. Not all genetic threads are bold. Many are fine and pale, only visible when the light hits just right. And then there are chromosomal disorders, when entire chapters of genetic information are added, missing, or rearranged. Down syndrome is caused by an extra copy of chromosome 21. Instead of 2, there are 3. That one extra chromosome affects development, facial features, and intellectual ability. It brings both challenges and uniqueness, an altered rhythm in the song of life. Turner syndrome, on the other hand, occurs when a female is born with only one X chromosome instead of 2. It can affect growth and fertility, another rearrangement in the Book of Instructions, but not all mutations are inherited. Some arise de novo, new changes that happen during the formation of egg or sperm, or just after fertilization. Spontaneous, random, a natural part of being alive. Imagine a scribe copying a book. After billions of copies, even the best scribe makes a mistake. Most are harmless, some are helpful, and a few cause the ink to blur. You may be a carrier of a mutation without knowing it. You feel healthy, you live fully, but tucked in your DNA is a silent variation, like a sealed envelope in your pocket. If your partner carries the same variation, and both of you pass it to a child, the message inside may unfold. This is how recessive diseases appear, only when two silent threads align. Tay-Sachs disease is one such illness. It affects the brain's ability to break down certain fats. Symptoms begin in infancy, vision dims, muscles weaken, life becomes fragile. It's heartbreaking, but it's not fate, it's biology, without blame. Still, even in these tangled patterns, there is resilience. The same sickle cell mutation that causes disease when inherited from both parents can protect against malaria if inherited from only one. Nature balances. What brings risk in one environment may offer strength in another. Mutations tell stories, not just of illness, but of survival. Your ancestors lived through drought, disease, famine, war. Their bodies adapted. Some of those adaptations now live in you. You are both their memory and their experiment. But inheritance is not the only author of your story. Environment plays a part too. What you eat, where you live, the air you breathe, these can shape how your genes behave. This is the field of epigenetics. If genes are the script, epigenetics is the lighting, the music, the stage direction. It doesn't change the words, but it can silence a scene or spotlight a phrase. Stress can alter gene expression. So can joy. Trauma can leave molecular bookmarks. And sometimes those bookmarks pass down. Your grandmother's hunger, your grandfather's fear. They may echo in your cells even if you've never known their story. But remember, epigenetic changes are reversible. Bookmarks can be moved. New choices can shift the narrative. You are not locked in. Your DNA also carries ancient scars. Remnants of viruses that once infected your ancestors' cells. Some embedded themselves into the genome, becoming endogenous retroviruses. And now, strange as it sounds, some of these viral genes help you. One assists in placenta formation, another helps detect threats in the immune system. What was once an invader is now an ally. You are not only your parents' child, you carry threads from creatures that once tried to harm humanity and were woven in instead. So tonight, as you rest, let this thought drift close. Your body holds errors and echoes, tangles and grace. You're not made of perfect lines, but of living patterns. If you carry a genetic disease or a risk, or even just the question, know this: you are not broken. You are not wrong. You are written in a language still being learned. And as science learns, it begins to whisper, could we untangle the threads? Could we find the knots and smooth them? Could we correct the code, not just for you, but for generations to come? The first steps have already begun, with tools like CRISPR, with therapies that insert healthy genes, with newborn screenings that catch illness before symptoms start. We begin to dream of mending the thread, not just watching it unravel. The story of inherited disease is not one of despair. It is a story of precision, of courage, of healing. And you are part of that story. Then even in tangled threads there is beauty. Even in broken places, there is light. So rest now with sound, soft hope. In the next chapter, we follow the scientists, the gene whisperers, the code editors. Who dare to ask? What if we could fix it? What if we already are? Outside, the world is still, but inside your body, a gentle choreography continues. Steady, quiet, unseen. You may not feel it, but right now your cells are awake. They hum softly with purpose, tending to their work like lantern-lit caretakers. In the great cathedral of you, this chapter belongs to them, to the hidden editors, to the quiet menders of life. Inside every cell in your body lies a copy of your DNA, your instruction manual, your personal blueprint. But this code is not carved in stone, it is delicate, vulnerable to time, chance, and outside harm. Sunlight, radiation, pollution, even the food you eat. These can sometimes nick the strands, twist the helix, or change a single letter in the script. Even normal life. The simple process of copying DNA before cell division can lead to tiny mistakes, but you are not defenseless. You were born with editors, molecular caretakers who roam your cells, reading the code, searching for errors, and gently making repairs. Imagine a librarian walking through a library of mirrors, each book a genome, each page made of living thread. That librarian is your DNA repair machinery, a group of proteins and enzymes that scan your DNA with unwavering focus. Their job is simple: keep the story clear, fix the smudges, replace the broken words, mend the pages before the meaning is lost. There are many kinds of damage, and for each a different repair. Sometimes a single base is wrong, a letter A where a G should be. This is called a point mutation, like a typo in a poem. To fix it, your cell uses base excision repair, a system that snips out the incorrect base and replaces it with the correct one, as easily as swapping a puzzle piece. Other times, a few bases are mismatched, slipped during copying. This is caught by the mismatch repair system, which runs its fingers along the strand, detecting bulges and misalignments. It straightens them out, like smoothing wrinkles from silk. But the most dangerous damage is the break. Sometimes a strand of DNA is cut, clean through like a snapped rope. These are double-strand breaks, and they can be devastating. Left unrepaired, they can disrupt genes, or even lead to cancer. So your cells treat them as emergencies. One way they respond is with non-homologous end joining, a fast but imperfect repair where the broken ends are stitched back together, even if small pieces are missing. It's like patching a torn sail mid-storm. It may not be perfect, but it keeps the ship from sinking. Another, gentler method is homologous recombination. Here, the cell looks for a backup copy, often from a sister chromatid, and uses it as a guide to rebuild the break, exactly as it was. It's slower, more precise, and closer to true healing. This process is especially important in cells that divide quickly, like bone marrow or skin, where mistakes could multiply. You might be wondering, if these systems are so powerful, why do diseases still happen? Why doesn't the body fix everything? The answer lies in balance. DNA repair is incredibly effective, but it isn't perfect. Your cells are constantly under stress, and over time repair systems can wear down. Sometimes the repair enzymes themselves are damaged, mutated so they no longer work. When that happens, errors build up. If those errors occur in genes that control growth or division, cells can begin to multiply too fast, refusing to die when they should. This is how cancer can begin. Not in a single dramatic moment, but in the slow failure of editing. Some inherited conditions come from problems in the repair systems themselves. Take xeroderma pigmentosum, a rare disorder where people can't repair UV damage to their DNA. To most of us, sunlight is warm and welcome, but to those with this condition, even a little sunlight can harm the skin's DNA and raise the risk of cancer dramatically. There's also Lynch syndrome, an inherited fault in mismatch repair genes. It doesn't cause cancer directly, but increases the risk of colon and other cancers by allowing more mutations to accumulate unchecked. These conditions show how vital the editing systems are, and how fragile, but still, the story is not one of despair. Even when repair fails, your body has layers of protection. Cells with too much damage often undergo apoptosis, a programmed death. They fold themselves quietly, gracefully exiting, so they do not pass the damage on. It's a last kindness, a selfless gesture of protection. There is beauty in this orchestration, this unseen ballet of maintenance and repair. You live because trillions of edits happen every second. As you breathe, your DNA is watched over. As you sleep, the errors of the day are mended. It is the body's form of nightly housekeeping, tidying up the code, so that tomorrow you may begin again. And in this way, healing begins long before we even notice. This constant care has also inspired something new. Scientists study your cell's natural repair systems to learn how to fix genetic diseases more precisely. They ask, if the body can edit itself, can we support it? Can we guide the repairs, or trigger them in cells that need help? This is where the future of therapy begins, not with invention, but with imitation. Learning from nature's editors, so we can become editors too. Some treatments already use this idea. Take gene therapy, where scientists insert a healthy copy of a gene to replace or override the faulty one. This doesn't delete the original, but adds a new script beside it. In some cases, like with sickle cell disease, gene therapy works by prompting the body to activate a gene that had gone quiet, a backup that can take over the job. It's not just repair, it's reawakening. CRISPR too relies on the body's own repair tools. It makes a precise cut in DNA, then lets the cell repair the break, either by pasting in a healthy gene or silencing a harmful one. The magic is not only in the scissors, but in the repair that follows. Nature becomes the partner, not just the patient. So now, as you rest, know this. Inside you there are caretakers. Not just doctors, not just researchers, but molecular guardians. They work silently, faithfully, every night of your life. They mend, they restore, they protect. They do not seek thanks. They do not sleep. And even when they falter, science listens. We watch what they do so that we may help when they need help. In this, there is a great and gentle hope. So let your breath soften now. Let your thoughts wander. You are held not only by blankets, but by intricate systems of trust that live inside you. You're being mended in ways you will never fully know. And just ahead, we step into the next circle of this story. Now that we've seen how the body edits itself, we will explore how science joins in, how medicine begins to rewrite the code with care, to ease suffering and rewrite possibility. The thread once broken may yet be whole. Sleep with that thought. The next chapter waits in quiet hope, deep in the hush of night, wrapped not just in fabric, but in quiet wonder. Inside you, cells dream of order, of clean instructions, of smooth threads unbroken. But when a letter in your genetic code is misplaced, the dream can falter. And so the body tries, as we saw before, to mend its own instructions. Yet sometimes the damage is too complex, too quiet, or simply too hidden to find. And that's when science begins to whisper alongside nature, offering a hand, offering a tool. Tonight, you'll meet that tool, delicate, precise, like a pair of invisible scissors, held by the gentlest fingers. Its name is CRISPR. CRISPR is not human in origin. It comes from the smallest life forms. Bacteria, those ancient, humble guardians of their own tiny worlds. Long ago, these microbes faced invaders, viruses that tried to hijack their machinery. And so in their defense, they evolved something extraordinary, a memory. They learned to save pieces of viral DNA, storing them like mugshots, in a genetic scrapbook. And when that virus came back, they used these records to recognize the threat and send out a cutting tool to destroy it. That cutting tool is an enzyme called Cas9. Together, this system, CRISPR, the memory, and Cas9, the scissors, forms one of the most elegant defense strategies in all of biology. Not brute force, but memory and precision. Now imagine this. Scientists watching bacteria realized what they had discovered. A way to guide a molecular blade to any point in the genome. All it needed was a map, a short guide RNA, that matched the sequence they wanted to edit, like giving coordinates to a surgeon who could then walk into the body's library and open exactly the right book on exactly the right page and snip a single word. Not with clumsiness, but with intention. The elegance of CRISPR is in its simplicity. All genes are made of 4 letters, A, T, C, and G. And CRISPR can be told with startling ease which letters to find and where to cut. It doesn't rewrite on its own, and it makes the break. Then the cell takes over, trying to repair the break, just like it would with any natural injury. And here is where the magic deepens. If we give the cell a new template, a corrected version of the gene, the cell may use that template to rebuild what was broken. This is called gene editing. Not just cutting, but rewriting, like offering a new lyric to a song that had gone off key. CRISPR has already changed the world, not just in theory, but in practice. It has been used to treat diseases, once thought unchangeable. Sickle cell disease, and a painful inherited blood disorder, was one of the first to feel CRISPR's touch. Scientists found a way to turn back on a fetal version of hemoglobin, a protein babies use before birth, but usually switch off later in life. By editing a control gene, they let the body keep making that safer form of hemoglobin. And in some patients, the pain vanished. CRISPR isn't magic. It has limits and risks. Sometimes the scissors cut in the wrong place, called off-target effects. And that can cause unexpected problems. Sometimes the cell doesn't repair the break the way we hope, and sometimes the editors don't take in every cell, especially in adults, where cells are already spread and specialized. But researchers are learning, refining, watching nature's hand at improving their own. They have even created new versions of CRISPR, gentler ones that don't cut at all. One of these is called base editing. Instead of breaking the strand, it changes a single letter, without snapping the thread. Imagine a pencil eraser rubbing out a single wrong character, and penciling in the right one. It's slower, more patient, and perhaps even safer. Another is prime editing, which acts like a word processor able to search, delete, and replace text with careful strokes. The rise of these tools shows how science continues to soften, to refine, to listen more closely to the language of life. There is power in this, but with power comes a pause, because CRISPR doesn't just fix disease. In theory, it could change almost anything. Eye color, height, even aspects of personality, though that remains a vast and uncertain field. These are not just repairs, these are choices, and choices ripple. When scientists edited human embryos in 2018, not for a deadly disease, but to change a potential future child's resistance to HIV, the world trembled. The experiment was condemned, not because the science failed, but because the ethics did. It was too fast, too soon, without full understanding. It was a reminder. Just because we can does not mean we should. CRISPR brings us to the edge of something vast, a future where inherited diseases might be corrected before birth, a world where suffering, once passed silently through generations, could be softened or erased, but also a world where editing the human genome must be approached with reverence, because every change is a message to the future. You resting now are part of that future. And though CRISPR lives in labs and test tubes, its heartbeat echoes in you. The hope that one day a child born with a harmful mutation might be spared its effects. The dream that someone need not carry the same burden their parents did. This isn't fiction. It is already happening. But slowly, carefully, as it should. And CRISPR's power doesn't end with humans. It's being used in agriculture, to make crops more nutritious, to help plants resist drought and disease, in animals, to study disease or even prevent extinction. There's talk of editing mosquitoes so they can't carry malaria, or modifying pigs so their organs might one day be used in transplants. It is the beginning of a long and winding road, but always we return to the beginning, to the simplicity of it, a strand of DNA, a small guide, a protein called Cas9, a clean cut, and then a choice. Will we repair the break? Will we rewrite the story? Or will we listen, wait, and learn? CRISPR doesn't answer these questions. It simply offers the possibility. Tonight, as you fall deeper into stillness, let this possibility rest with you. That life, once written, is not beyond revision. That nature, when hurt, can teach us how to heal. And that sometimes the smallest tools carry the gentlest hopes. In the next chapter, we step beyond the cell. We watch how scientists deliver these tools to living bodies. The challenge of reaching the right cells at the right time, in the right way. Because a tool must be guided, and the delivery is its own kind of miracle. Let your mind drift toward that chapter. Let the scissors rest. The edit can wait. For now, there is only peace. As you lie here, still and calm, feel how your body moves in subtle ways. Not with effort, but with rhythm. The pulse of your blood, the quiet whispers of your cells. Your body is a garden of cells, growing, shifting, or responding to the seasons of life. Roots deep in the soil of your DNA, branches stretching through the air of your thoughts, each part of you. Every nerve, muscle, bone, and organ is shaped by the soil from which you've come. The seeds planted by your ancestors grew into you. But what happens when the soil itself is troubled? When the seeds are flawed or sick? When a garden once rich begins to wither? This is where gene therapy comes in. A quiet, patient gardener who seeks not to fix the symptoms, but to plant new life at the very root. Gene therapy is a whisper to your genes, a whisper that says, let's try again. Imagine for a moment that your body's DNA is a library of all the instructions that make you, you. Every gene is a book, every sequence a chapter. Now imagine that one book is flawed. Its instructions are misprinted. And though the rest of the library is fine, this one book causes trouble. It causes sickness. It causes pain. Gene therapy is like a librarian who doesn't just patch up the pages, but finds a way to rewrite the book itself. Let's walk through how this happens. Gene therapy isn't a single solution. It's many solutions, crafted carefully for different needs. For some conditions, scientists take the gene that's broken, copy it, and replace the faulty version with a healthy one. It's like changing a single page in the library, one letter at a time until the instructions are correct. In other cases, the therapy works by inserting an entirely new gene. That helps the body perform a task it couldn't do before, like teaching the cells to make a protein they've never known how to make. The trick is delivering the new gene, getting it past the walls of the cells to the very heart of the DNA library. Imagine your body as a vast, bustling city. Every cell is a small building, every building full of workers carrying out their jobs. But in some of these buildings, the workers are following faulty blueprints. Gene therapy acts like a delivery truck, bringing new corrected blueprints to those buildings. The truck arrives, and inside a new set of instructions is carefully carried in. But not all the buildings are easy to reach. Some are deep within the city, or hidden behind walls, so the delivery truck must have a way to get in. This is where the real magic happens. Scientists use viruses, not the kind that cause illness, but harmless ones, to deliver the new genes. Viruses, you see, have an amazing talent. They know how to enter cells. Like clever messengers, they transport the new healthy genes deep into the body, delivering them precisely where they're needed. And once inside, the new gene can do its work, repairing, replacing, or adding to the instructions that guide your cells. Let's take a step deeper into this world. The first real success of gene therapy came with severe combined immunodeficiency, SID, commonly known as bubble boy disease. Children born with SCID have an immune system so weak they cannot fight off even the simplest of infections. It's a devastating disease passed down through genes. But in 1999, scientists began using gene therapy to help these children. They took cells from the children's bone marrow, added the missing gene, and then returned the cells to their bodies. And slowly their immune systems began to work. These children, once isolated, living in sterile environments to protect them from the outside world, were given a chance to play outside, to live freely. There are more stories quietly unfolding. One of the most remarkable is the story of Lux Turner, a gene therapy used to treat a rare form of inherited blindness. For patients born with Leber congenital amaurosis, their retinas, the light-sensitive layer at the back of the eye, fail to function because of a missing gene. In 2017, Lux Turner became the first FDA-approved gene therapy for an inherited condition that causes blindness. A single injection in the eye was all it took. The new healthy gene was delivered by a virus, and over time patients began to regain their vision. For some, the improvement was dramatic. They could see faces for the first time or read words they hadn't read in years. There is also the case of Zolgensma, a life-saving therapy for children with spinal muscular atrophy, SMA. SMA is a genetic condition that robs children of their ability to move, and it's one of the leading genetic causes of death in infants. But Zolgensma delivers a working copy of the missing gene, giving the child's cells the ability to produce a protein that is essential for the survival of motor neurons. It's a single-dose therapy, and for many children, it has been nothing short of miraculous. It has allowed them to sit up, walk, and live in ways that would have once been impossible. These are stories of hope, but they are not the end of the road. Gene therapy is still young, still learning how to reach every cell, how to make its edits perfectly. Some challenges remain. Delivering genes to the right cells at the right time, in the right amounts, is a delicate process. Too much of a gene can be as harmful as too little, and the body can sometimes respond with a protective immune reaction, rejecting the therapy before it has a chance to work. But with each passing year, scientists get closer, and with every success, a new possibility is born. Perhaps the most beautiful part of gene therapy is the way it speaks to the future. In the past, we could treat disease only after it showed itself. We could manage symptoms, but not prevent the storm. Gene therapy offers the possibility of changing that. It whispers to your genes before the disease has a chance to take root. It's as though instead of waiting for the garden to wilt, the gardener walks in and plants new seeds that can grow into stronger, healthier plants before the old ones have time to wither. And so as you rest now, think of your body as a garden, one that has weathered storms and yet still stands. In your cells there is more than healing. There is potential. Imagine, as you drift deeper into your peaceful rest, that the future holds a quiet promise. A promise where diseases no longer simply exist, but are rewritten. This is the garden that is being cultivated now, not just with medicine, but with the gentlest touch of science in the form of gene therapy. And though the road is still unfolding, the seeds have been planted. In the next chapter, we'll turn our attention to the questions that remain. What happens when we edit the genome, when we change life's script? And most of all, what does it mean to rewrite a future? Let your mind rest with these thoughts, like seed sown in the quiet night. The night around you is soft and deep, and your breath moves gently in and out, in and out. As you drift in the quiet, imagine something delicate and something profound. A moment before the first breath, before you ever opened your eyes, before your heart learned to beat, there was a time when you were nothing more than a promise. A single cell, just one, growing and splitting and growing until the intricate tapestry of your body began to form. And from that first whisper of life, everything about you began to take shape. the most delicate moment of your life, was when you were the youngest you'll ever be. The single fertilized egg, the zygote. The earliest cells dividing in perfect harmony, holding all the blueprints. That would become you. Now imagine that we could touch those cells. Imagine, before your first breath, before your body ever learned to grow, we could edit your genes. What could be possible then? At this moment, when cells are still young, they hold incredible potential. The power to become anything, to form anything. This is the magic of early editing. When we alter genes in these first cells, we change the very foundation of life, the first seed from which a person will grow. The younger the cell, the easier it is to guide it, to whisper a new story into its DNA. In these early moments, the cells are like clay waiting to be shaped. This is why when we dream of changing genetics, we often look to the younger cells, those that are still undivided, still pure in their potential. The potential for change is highest then, because those cells, those first few, will multiply to become every part of you. Your heart, your lungs, your skin, the cells we alter early will carry that change through every fibre of your being. Now pause with me for a moment. Think about this question quietly in your heart. What does it mean to change a child before they take their first breath? There is something deeply profound in this idea, a weight to it. When we change genes in these earliest cells, we aren't just changing a body. We are shaping a future, a life that has yet to be lived, a mind that has yet to form. If gene therapy holds the power to heal, it holds a responsibility too. It is one thing to heal a body already in motion. It is another to alter the course of a life before it has even begun. Let's step into this world carefully. Imagine a baby in the womb. From the moment of conception, the baby's cells are dividing, multiplying, and taking shape. This is the moment when gene editing can have its most profound effect. When the cells are still blank slates, unwritten by time, the earlier we begin to edit, the more the changes ripple outward, shaping the child's body, their health, and even their future children. This is not just about preventing diseases, it's about altering the path of a person's life before it's even begun. For example, imagine a child at risk for a genetic disorder, a disease that runs in their family, a disease that could change the course of their life. What if before birth we could edit the gene responsible for that disease, removing the faulty blueprint and replacing it with a healthy one? This would not be a cure in the traditional sense, it would be prevention. It would be rewriting the story before it begins, and in doing so we might eliminate suffering before it has the chance to even exist. Yet with this power comes a question. Where do we draw the line? If gene editing can erase disease, can it also enhance? Can we improve a child's traits, their intelligence, their strength, their beauty? The question becomes deeper then. If we can edit genes, how much should we? Should we edit just to cure, or do we have the right to shape and to enhance? This is the very question that haunts scientists, ethicists, and all of us who dream of a better future. But there's another way to think about this. A gentler way. What if we think of gene editing not as a tool of control, but as a tool of healing? Healing is not just about fixing what is broken. It's about preventing harm, about giving the child the best possible start in life. Imagine, for example, a child born to parents with a history of heart disease. What if we could edit the genes to give that child a heart that was stronger, less prone to disease? Imagine a child born with the genes that might cause cancer one day. What if we could edit those genes before they even had a chance to take hold? In this world, gene editing isn't about perfection. It's about compassion. It's about giving life the best possible chance to flourish, without the weight of inherited burdens. But even in this quiet, gentle world, there are risks. There are uncertainties. We do not fully understand all the consequences of gene editing. What happens when we make changes so early and so deeply? Could altering the genetic code at the start of life lead to unforeseen problems down the road? The answer, as it often is in science, is that we don't know yet. We are standing at the beginning of something new, something powerful, something full of hope, but also full of mystery. But still, there is a quiet beauty to this potential. Gene editing is not about controlling life. It's about offering it the chance to be free from suffering, free from inherited diseases, free from the limitations placed upon it by the past. And as we gaze into the future, we wonder, could we one day begin life with a blank slate, free from the weight of inherited burdens? Tonight, as you drift further into rest, carry with you the weight of this question. What does it mean to begin again? The story of life is more than just one person. It is the story of all of us, and with gene editing, we may be learning to write a new chapter. In this ongoing story, a chapter of hope, of healing, of beginnings. In the next chapter, we will consider the ethical landscape of this journey. How do we decide what is right? What does it mean to have the power to shape life itself? But for now, let your thoughts rest. Let them wander like seeds in the soil waiting to sprout. The night is still and your breath moves slow, steady. Tonight we step into a quieter place, a place not of science alone, but of thought, of questions that linger in the air like the scent of rain before it falls. Imagine this. We have the power to change life, to rewrite the code that makes us who we are, to edit the very fabric of being. A child's future, a body's health, a mind's potential, they could all be touched, altered, rewritten. But with this power comes a question so profound it hums through the world like a song. We can't quite finish. Should we change? In this quiet in-between space where we pause, we listen to the echoes of possibility. Because the question isn't just can we, it's should we? And when we ask that, we find ourselves in the grey. The world of gene editing and of altering the very blueprint of life is not a world of simple answers. It is filled with layers, with nuance, with complexity. A place where help and harm can look so much alike, where cure and control dance together, in quiet balance. We begin by considering the scope of change. When we edit genes to eliminate disease, we are performing a kind of healing, a powerful, necessary healing. But healing is not always a clear path. Sometimes healing means removing the past. Sometimes it means eliminating suffering. But sometimes healing can also mean controlling the future. Imagine a world where we can choose traits, the colour of our child's eyes, their height, their intelligence. Imagine a future where parents can select the ideal genetic combination, as if choosing the pieces of a puzzle, placing them with precision. Does that sound beautiful? Does it sound right? It is tempting to say yes, but in this world of perfect control, where would we leave room for the randomness of life? For the unexpected joys that come from things we cannot plan? For the quirks that make us who we are? The imperfections, the surprises, the things we never saw coming? But that turned out to be the most important. And what of equity? If gene editing becomes accessible only to some, then how would we divide the future? Would it be divided into those who can afford perfection and those who cannot? If we can create a generation of perfect children, will the pressure to be perfect become too great? Will our definitions of health and success be narrowed so that only one path is acceptable? One path is worthy? These are the questions we must ask, even as we stand on the edge of possibility. Yet there is a gentler question to consider. Can gene editing be used responsibly? Can it be focused only on healing, on relieving the burden of disease? What if, instead of editing genes to create a world of uniformity, we focus on curing diseases that cause suffering? What if we use this power not to control, but to eliminate suffering where we can? This is the power of gene therapy, the power to heal not just a body but a future, to cure inherited diseases before they can take root, to give children the chance to live without the burdens of their parents' genes. There is beauty in this. There is hope. But there are shadows too. What happens when we start to define health only in certain terms? When we begin to treat disease as something to be eradicated, and health as something to be controlled? The more we change, the more we risk losing the very thing we are trying to protect. The natural, unpredictable dance of life. The complexity that makes us human with all our imperfections, our uniqueness, our diversity. Pause now and feel the weight of this question. What future do we want? It is not just a question for scientists or doctors. It is a question for all of us. If we have the power to change the very building blocks of life, how do we decide what is worth changing, what is worth preserving? Perhaps this is the most profound question of all. What does it mean to be human? We come back to this space, the space where we can edit the human genome, where we can alter the very essence of life. But what does it mean to alter it? What is worth changing and what must remain untouched? Is the answer different for everyone? Can we agree on what is right, or will the debate continue, echoing across generations? As we step back from the edges of science, we must ask the deeper questions. Questions of ethics. What is our responsibility when we hold the power to change life itself? And when we stand on the brink of this new world, we realize that this is not just a scientific question. It is a human question. Some believe that gene editing is the next step in our evolution, that it is our responsibility to cure disease and to eradicate suffering, to make a world where every child can be born healthy and strong. Others wonder if we are crossing a line, if we are stepping into a realm where we do not belong, where in trying to perfect life we risk losing the richness that comes from being human. And there are those who ask, what if the changes we make today will have consequences we cannot foresee? What if in editing the code of life we create new problems we are not prepared to handle? But still, even as these questions echo, there is hope. Hope that we can move forward with care. Hope that we can use our power for good, to heal, to ease suffering, to make the world a better place, without losing what makes us whole. As you rest tonight, let these questions drift with you, as the night holds them softly. In the stillness, find peace in the uncertainty, knowing that these questions are part of the journey. For in the echoes of these questions, we are not just asking what to change, we're asking what it means to be human, to care, to heal. And in that asking, we find the future. In the next chapter, we will explore the possibilities ahead, the hopes, the dreams, and the reality of what's to come. But for now, let these quiet questions rest in your heart as you drift gently to sleep. As your body rests, your mind drifts deeper, not just through time, but across generations. What if, in the stillness of this moment, you could reach back and touch your ancestors? What if you could reach forward and touch the future? Tonight we consider a delicate possibility, a change, not just for today, but for all the tomorrows to come, a change that ripples outward, spreading its gentle currents across generations. We are no longer just rewriting the stories of our lives, but the stories of lives yet to be born. Imagine this. You are holding a thread, a single thread of life. It is the thread of your genes, woven deep into the fabric of your being. And what if, with a careful snip, you could alter that thread? Not just for you, but for everyone who will come after. This is the quiet power of germline editing, the ability to change the very genetic code passed from one generation to the next. A change that does not end with you, but that continues quietly and steadily through time. Germline editing is the art of reaching into the beginning, the moment when life begins to form. It is the art of changing not just your own body, but the blueprint that will be handed to your children, and your children's children for as long as the lineage endures. But this gift, this power, is not without its weight. To edit the germline is to alter something sacred. It is to reach into the very core of life's future, to touch the invisible threads that bind generations together. The question that echoes through this delicate space is, what right do we have to change the path of future lives? You are standing at the crossroads, where science and ethics meet. In one hand, you hold the possibility of healing, of eradicating diseases that have plagued families for generations. You hold the promise of children born without the burden of inherited illness. And in the other hand, you hold the uncertainty, the unknown consequences, the ripples that may stretch far beyond what we can predict. This is the heart of germline editing, a choice to edit not just one life, but the lives of many, the lives of all who come after. It is a choice that lingers in the quiet spaces between what we can do and what we should do. Imagine now a family where a genetic disease has been passed down through the generations. It is a disease that causes suffering, a pain that ripples from one life to the next. Every generation bears the weight of this invisible burden, and no matter how much medicine advances, it never quite disappears. Now imagine a world where this burden could be erased, where the very DNA that carries the disease could be altered before the child is even born. A world where the future of this family and all the generations to follow could be freed from the grip of disease. There is beauty in this, and hope, a life without the shadow of inherited illness. A world where every child is born with a clean slate, free from the mistakes of their ancestors. But this change is not as simple as it seems. For what does it mean to erase the past? What happens when we decide that a certain condition is something to be avoided, to be erased? In this new world, what becomes of the diversity that makes us human? What becomes of the imperfections, the quirks that define each person? What happens when we begin to decide what should and should not be passed on? When we start to make choices about what parts of humanity are worth keeping, and what parts should be rewritten? These are the quiet questions that germline editing brings with it. Questions of control, of power, of the future we are shaping. And there is another question too. What responsibility do we have to the future? A single change in the genome today can echo across generations. It is a change that lasts not just for a lifetime, but for as long as the lineage endures. What will the children of tomorrow say of the choices we make today? What will they think of the legacy we pass on? In this moment, we are not just editing genes, we are editing futures. We are shaping the lives of those who will never speak our names. We are laying the foundation for their world, their bodies, their minds. As we consider these questions, we must pause, for the power to change the future is both beautiful and terrifying. It is a power that comes with both great promise and great responsibility. What does it mean to change a future before it is born? What does it mean to decide for someone else what their life will be like before they have the chance to live it? In the quiet space between these questions, we can hear the faint echoes of our compassion. For surely if we are to change the future, we must do so with compassion, with grace, with humility. We must ask ourselves, not just if we can change life, but if we should. And if we do, we must do so with the greatest care, knowing that the ripples we create will spread far beyond what we can imagine. Now, imagine a world where compassion is woven into our legacy, where the choices we make today are made with the future in mind. A world where the changes we make are not just for our own benefit, but for the benefit of all generations to come. This is the world that germline editing promises, a world where healing and compassion are passed on and woven into the very fabric of life. But as we step forward into this new reality, we must carry this responsibility with grace, with humility, and with the understanding that we are not just shaping one life, but many. As you rest tonight, let these questions settle in your heart softly and gently, for in the quiet spaces between them, we find the true weight of what it means to heal, to change, and to love the future we are shaping. Tomorrow we will explore the deeper consequences of these choices, but tonight we rest in the mystery and in the beauty of what it means to carry the weight of change with compassion. Science dreams in laboratories and hearts. We glide into tomorrow's questions, safer edits, broader cures, gentle revolutions. There is no final chapter to healing, only a steady unfolding. You rest now knowing that your body, your future is full of wonder. As you rest tonight, let these questions settle in your heart, softly and gently. For in the quiet spaces between them, we find the true weight of what it means to heal, to change, and to love the future we are shaping. Tomorrow we will explore the deeper consequences of these choices, but tonight we rest in the mystery and in the beauty of what it means to carry the weight of change with compassion. Now we drift into the last pages of this quiet journey, a journey not toward a destination, but into the forever unfolding of possibility. Because even as you sleep, science does not rest. It moves gently like moonlight on water, spreading across the world. In the flicker of a microscope and the silent click of day being read, in the hush of a scientist's breath as they glimpse something new, the dream of curing inherited disease is no longer just a dream. It is a path we are walking step by delicate step, with bare feet and careful eyes, not with haste, but with reverence. The unfinished symphony. Imagine your body again, that silent orchestra of cells playing your life song. Each note, each gene, tuned by time, by ancestry, by chance. Sometimes a note wavers, sometimes the music falters, and yet the melody continues because life, even flawed, is persistent and beautiful. What science offers now is not a new song, but a way to retune the broken strings, to coax the music back to its intended rhythm, to allow every person the chance to play their song, clearly, fully, freely. But this is not a single fix, not a universal key. This is a symphony still being composed. Beyond CRISPR. CRISPR was only the beginning. Since its discovery, scientists have dreamed of even finer tools, like tiny quills writing letters in water, or silk threads sewing seamlessly through the very fabric of life. One new approach is called base editing. It doesn't cut the DNA, but instead it gently swaps one letter for another, like correcting a single typo in a book, without tearing the page. Another, prime editing, offers even more precision, as if the cell is handed a thoughtful rewrite of a sentence and asked kindly to consider replacing it. These new techniques are softer, subtler. They reduce the risks. They whisper to the cell instead of shouting. And that's the direction science is leaning toward now. Not just fixing what's broken, but doing so with care, with elegance, with a humility that matches the complexity of life itself. Listening before speaking. As we gaze softly into the future, science is learning something else too, to listen more closely to the body before trying to change it, to understand the entire ecosystem of a gene, not just what it says, but who it talks to when it speaks, and how loud its voice becomes. Because genes do not act alone, they speak in chorus, and altering one part can echo through the others. Ripples in a pond, reverberations in a song. So the future is not only more powerful, it is more respectful, and perhaps the most profound shift of all is that scientists are beginning to ask the body what it needs, instead of assuming they already know. Hope for all. Right now, most gene therapies are rare and expensive. They reach only a few. But the dream, the quiet revolution, is that one day these tools become simple, affordable, available to anyone who needs them. To a child in a quiet village, to a parent watching and waiting by a bedside, to a community long overlooked, to everyone equally. Not because they are rich, but because they are human. That future is not here yet, but it is closer than ever. Some scientists are building tiny gene therapy kits that fit in a fridge. Some are learning how to deliver treatments with a puff of air or a swallow of liquid. Some are working on one-time cures that last for a lifetime, because healing shouldn't be rare. It should be ordinary, gentle, accessible, like breath. The new healers. And in this future, you are not only the healed, you are also the healer. Your voice, your questions, your choices matter in shaping what comes next. Because science is not separate from you. It lives in your hands, your stories, your values. More and more, communities are being asked to guide research and to say, this is what we need. This is where we hurt. This is how to help without harm. And slowly, softly, the labs are listening. Science becomes not just a profession, but a conversation, a relationship, a shared lullaby written by many voices, what healing means. As we close our eyes now, we return to the deepest question. What does it really mean to be healed? Is it the absence of pain? Is it the fixing of flaws? Or is it something more mysterious, a return to wholeness, even when not everything is perfect? Sometimes healing is not about changing your genes, but about changing how the world sees you. Sometimes it's not a fix, but an embrace. The future of science may offer both, the power to mend what hurts, and the wisdom to honor what already shines. You are not broken, you are becoming, and science, when guided by care, can help you bloom more fully into who you already are. The stars keep turning. Look up, in your mind, to a sky full of stars. Each one is a possible future, a life that could be healed, a child born without the burden of disease, a parent who no longer worries, a world that chooses compassion as its North Star. We will not reach them all, but we can choose which ones to steer toward, and every step, every question, every whisper of hope is part of the voyage. The work will go on. The cures will come slowly and then all at once. But you here, resting now, are already part of the story. You carry the dream just by believing it is possible. A soft goodbye. So now we close this chapter, not with an ending, but with an open hand. A quiet promise that healing is not a finish line, but a path walked daily. By researchers, by patients, by dreamers like you. As you drift into sleep, remember, the body holds stories, the cells remember hope, and the future, however complex, can still be tender. May your dreams be full of soft revolutions, of codes rewritten with care, of tomorrow's children born with fewer burdens. Let the stars turn and let the genes hum, and let yourself rest. You are part of something vast, something beautiful, something just beginning. Good night.